Okay.
Live. This is like, do you remember the Gene Machine that we did last year, the skit?
Oh,
yeah.
That was fun. You remember when you like make a skit about like, I don't know what it was about, but like each group made their own skit.
I don't even remember.
No.
Like, remember when we did it and you don't remember? You know what I'm talking
You know what I'm talking about.
I know.
That thing. All right. Well, I'm waiting for, yeah, we're waiting.
Oh, he's
We
not
don't
on
have
yet.
any viewers either. So.
Well, no, we don't want to, do we want any viewers?
No.
Yeah, let's just have public broadcast at GPHS.
Yeah.
Hi, this is GPHS. We're here to talk about algae.
So he'll show up in the corner here in just a minute.
So do you guys want to like rotate by team? Like we'll still
Yeah.
listen to your questions, but like once you guys are asked
Yeah,
questions, we
we
can
can
move up and ask.
No, we're
Well, all right.
Yeah.
Where I belong.
Ah, there he is.
Yeah.
Okay, good. It's pretty easy.
He's coming.
He's coming.
Hello,
I don't know. We think you're coming.
I'm here.
Okay. But we can't see you very well. There you are.
How do you make him bigger?
All
I want to know
right.
how to make you bigger. There you are.
Make him bigger.
Okay.
You got pretty backlit.
Oh, there you go.
Or, no, right.
Okay, I'm trying to get the volume up here. There. Are you there?
I'm there. I'm here. You guys.
Okay, well, I got a whole bunch. Can you see them? I'm just going to back out of the way. So they can introduce, they've got questions. I'm going to go out to the greenhouse and make sure nobody's electrocuting themselves, and then I'll be right back. This is Dr. Jake. So as you ask questions, introduce yourself, and they're yours.
Yeah, go for
Go for
it.
it. Who's that?
Hi, I'm Celine, and we have a few questions
Hold on.
about
Hold on.
algae.
Hold on.
So our thesis from my like experiment is if we add CO2 to the algae, will it increase the lipids? And
Wait, hold on.
if,
Stop first. You guys can hear me, right?
yes.
Okay, so there's a delay for one. So when you ask the question, wait for a response. The other thing is, let me introduce myself real quick.
Oh, okay.
Okay, you guys don't know who I am, right? Because so I'm a postdoctoral fellow at Institute for Systems Biology, and I've worked with Tammy for about 3 years. I got my PhD in Montana working on thermophilic cyanobacteria and turning diatoms into biofuel. So I have a lot of experience understanding the physiology and the systems biology behind that process. Okay, so and I also worked with a company a couple of years ago that actually was converting algae biomass into biofuel. So I have a lot of work with the academic side and doing the research, and the industrial side and how to convert that into biofuel. Okay, so feel free to ask all the questions you want, and you guys already kind of started. Introduce your name, what group you're on, and that kind of stuff, and we can start there. Okay? All right, so go ahead and start over. Sorry.
Okay, so we were wondering how much CO2 we should add to the algae, like at what range?
Okay, so that's a good question. So obviously you're asking that question because if you're growing the algae photosynthetically, they're going to be using light, CO2, convert that CO2 into carbon chains. So do you guys understand like what happens when you put CO2 in water? What happens?
The water has CO2.
It affects the pH.
The pH level becomes acidic.
Right. So what happens is CO2 dissolves into water, makes carbonic acid. That carbonic acid disassociates to bicarbonate. So mainly the organism is taking up bicarbonate. So there's a certain point in which you can add too much CO2, and then your pH will drop really low and is no longer beneficial to the organism. That being said, a lot of the industry, they actually use a lot of CO2. You guys know how much CO2 is in the atmosphere right now?
4.2 or point something with 4. 4
or something. 0.04%.
Why don't you guys look it up? But it's around like 0.04%, right? So there's not a lot of CO2 in the atmosphere, right? So what a lot of companies do is they'll pipe in factory CO2 or CO2 that's concentrated at around 1% to 5% CO2. That's a lot of CO2. And you guys know how much percent is oxygen, right? In the air?
40.
20%.
Funny.
But so the point being, there's a range, and you can actually go pretty high without it being critical for the algae. So if you guys have access to add it up to about 1 to 5%, you should be fine. Does that make sense? You can become limited on CO2 if you're just pumping in. Say you just took an aquarium pump and you pumped in air. You basically pumped in 0.04%. That's plenty CO2 for the organisms to grow. But sometimes when the culture is really, really thick, it becomes CO2 limited, and there's not as much CO2. And so what happens is the algae doesn't make as much lipid. So you want to make sure that if you're at a high dense culture, you want to increase the amount of CO2 you're giving to the organism.
Okay. And then like a follow-up question to that is because like, as you said before, if you put too much CO2, it becomes acidic. How can we manage the pH so it doesn't become really acidic to like hurt the algae?
So what would be your first inclination? What do you think? If you had to solve it without asking someone with a PhD, what do you think you would try and do?
Add something like a medicine or a buffer.
Buffer, right? That's exactly what a buffer does. So they teach you in chemistry class that a buffer basically buffers the pH drop or increase, right? So it's another kind of a molecule that reduces or the effect of protons or pH on your media. So if you want to add 5% CO2, you can put in a buffer like say tris or heaps or some kind of biological buffer that says, okay, it's going to be around 7.8 pH, but I can increase my CO2 without the pH dropping so much. Okay?
And then another question is, okay, so we're adding, so for like our experiment, we're putting balloons and putting like the CO2 inside the balloons. Should we put a balloon on our control?
Yes, what would you put on it? Put inside the balloon.
Just nothing, just a balloon to like
Oh.
keep it like, because we want to see.
So okay, go explain your experiment again real quick.
So our experiment basically is we're trying to increase the lipids by adding CO2, and we're putting like CO2 in balloons and putting it over the flask.
Okay. So you'll have, how many flasks will you have?
We have 11 and one including the control.
Okay, so describe the control to you. We're just going to have it be.
We're just going to put the algae by itself.
I'm sorry, I didn't hear you.
We're just going to have like just the algae growing by itself with no CO2.
So just needs to be growing on air. Yeah.
CO2.
Like a normal amount of CO2?
Should we put like amount of CO2 in the atmosphere, like the 0.4, the 0.4?
Well, what I would do is I would just say use ambient air. So take the balloon and take just an air pump it up, and put it over the flask like you would the other. What that does is it mimics the positive pressure you're going to have from the balloon into the flask, right? That way it's more of a control, but then you say the control is just ambient air, so it's at 0.04%. That makes sense?
Yeah.
Also, if you're going to do it, have a few controls, don't just have one. Replicates are really important in biology, right? Because if you see an effect, you want to make sure that effect is repeatable and not just because some random thing that happened. So you want to have multiple replicas.
And then my last question is, so we're like growing the algae right now, but like at what time would we move the algae from the growing flask and dilute the algae to like separate them into individual ones? Like when would we dilute the algae?
I think what you're asking is how big the culture needs, how
Yeah.
robust the culture needs to be.
So I'm trying to get a picture of this. So you're going to grow up algae altogether, and then you're going to transfer them into new flasks and put the balloons on those flasks to control the CO2 coming in, correct?
Yes.
Yes.
That's what they're doing.
So, and your question, right? Whenever you design an experiment, the question starts with, or your experimental design starts with what question you're trying to ask. And what I'm gathering is your question is saying, what would the difference? How would you have a different effect on lipid production based on the CO2 concentration given to that culture? Correct?
Yeah.
Yeah, that's great hypothesis or great question. Experimental design is good. What I would do is if you grow the culture up in your media. I don't know if you guys are using folds. I'm assuming you are. Yeah. What you would want to do is when they start making lipid, usually when they run out of nutrients, okay? So if you do it the way you're suggesting, what I prefer we would do is grow the algae up when it's in exponential phase, meaning that there's still nitrogen around, there's still phosphate around, there's still sulfate around. And then I would transfer those cells into media that has no nitrogen, no phosphate, no sulfate. Right? So what that does, now you start starving the organism for its nutrients, and it'll start making lipids. And then the only variable you're actually going to change is the air that you're putting in. Does that make sense to you guys?
Yeah.
So what's a suggestion for media that doesn't have those things? Do we need to make our own?
Yeah, you would have to make your own. It could be pretty simple if you just balance out the osmolytes in the media. So if you just make the media, just don't give it nitrogen, phosphate, and sulfur,
Okay.
and add the amount of potassium, chlorine, and sodium chloride. That's pretty much it.
Okay.
So they're going to be alive for about 3 or 4 days because that's when they're going to make their lipid.
Okay.
And then one more question is, how much lipid does it take to actually make biofuel? Like, when will we know, like, it has reached the point so that algae can actually make biofuel?
So that's kind of an open-ended question. So a lot of times in industry, they talk about percent dry weight. So they'll say this algae will make 30% dry weight. I mean, if you took that algae, you removed all the water from it, 30% of that biomass is going to be lipid. Okay. Typically, when you starve an algae, they do about 10 to 30% dry weight. Obviously, if you're trying to produce biofuel, you want to maximize that. But that comes at a consequence, right? So if you starve the algae for 6 days, the algae might make 40% lipid per dry weight. But if you starved it for 2 days, it might only make 20%. Either way, it's the same amount of fuel because it took 2 days to get to 20% and 4 days to get to 40%. Does that make sense? Mm-hmm. So it depends on in your efficiency practices what you really want to do. I think for biologists, what we're trying to do is to make it make 40% lipid in 1 day kind of thing, right? If you can increase or decrease the amount of time it takes to make a maximum amount of lipid, that's when you start making a really profitable biofuel. That's what we're trying to do. And that's what you guys are doing with these experiments, is trying to figure out what's the best way and most efficient way to make the lipid that doesn't take a very long time.
Okay.
Does that help answer your question?
Thank you.
Yeah. Good luck.
Okay.
Go ahead. All right. Introduce yourselves. And tell what your question is.
All right. So I'm Michelle, and we're for a separate group. So for our experiment, we're taking 2 strands of mutant algae, and we're going to cross them over to try to see if we can create offspring that have both of the mutations. So the mutations that we're looking at are, one has very long flagella, and one has a lipid mutation so that it produces more lipids naturally, and we're trying to see if we can cross them. We'll have an offspring that has longer flagella and has that lipid mutation, so that hopefully it will produce lipids more efficiently than the regular wild type. Okay, so one of our questions is, because we're going to have to be studying to see if the genes are crossing over, what's an easy way to kind of track the way genes are being passed in algae?
This is, while you're describing your experiment to me, it's very ambitious. So let me ask you a question first. How are you going to determine whether your offspring actually exchanged genes, or that your offspring is from the mating of those 2 strains compared to just being the offspring of the same strain?
Yes. So what we have is we set up 12 different flasks. So we have and there's just 2 of each, so it's really just 6. So we have 1 with just the wild type. We have 1 with just 1 mutation. We have 1 with just the other mutation. Then we have a cross of each of those 3 in order to equal 6. So
what do you mean by cross?
So we have a wild type mixed with the first mutant, a wild type mixed with the second mutant, and then the two mutants crossed together. And then we're going to compare them based on the controls, which is just the mutants alone, to see if there's actually like a difference in between. So we're hoping that'll enable us to see like if there's a difference between, like if they're actually crossing over the strands.
Right. The problem with this is that you're talking about a population of millions and millions of cells,
Yeah.
right? You're not going to know, like if you have an outcome where there's less production, you don't know if that's an offspring. That's going to be your main problem, is you don't know if it's an offspring. That's why when they do the genetics, they have to do molecular screens and they have to do all this kind of stuff to make sure that you're actually dealing with an offspring and not just the growth of one strain versus the other. You see the complication in your experiment.
Yeah.
Very difficult to assess that. Now, it's easy to assess whether you have a cross strain molecularly, but to do it with a population of cells is going to be very, very difficult. So your original question was, how would you monitor that? Well, you would monitor is look for those two genes that you know that are to be mutants of those genes with PCR reactions in your offspring strains. The problem is you're going to have a mixture of offspring that maybe have both mutants, and then ones that are just the same strains as their parent. They're just propagated. Does that make sense?
Yeah.
So do you see the trouble with that? That's going to be very difficult.
Yeah. So
So are your strains, is it like STA6? What is the lipid mutant?
It's a number.
yeah, we had a number for it. I think it
Ended
was,
53, didn't it?
yeah, it was 5163. It was
Off of
from,
clammy.org.
yeah, off of clammy.org.
Oh,
Okay.
a question off of that, actually. My name's Sakura, by the way. So when we first got our algae samples, we ended up, so we put them in, we did it carefully, one by one, using sterile equipment and everything. And then what ended up happening was mold kind of appeared in the flasks in
Yeah.
each of the in the algae, and it also appeared inside the container that it was in. Do you happen to know how we can prevent like mold kind of appearing? Yeah. Because it just kind of, we had to start from scratch again. So.
Right. Mold is tough. Mold or fungi, they make spores, and a lot of times these spores are smaller than cells, so they can get through certain membranes and stuff like that. The biggest thing is to autoclave your equipment, autoclave your flask, your media, whatever it is that is going to get in touch with the cells. You want to make sure you can denate or destroy anything that's in there. When companies or institutions grow algae in their growth chambers, we usually have multiple strains. We have up to 100 strains of algae. One of the biggest things we prevent is mold, because that is the hardest one to get rid of, and it's the, hold on, my battery's getting really low.
Did you have more questions?
Give me 1 second.
I think he's busy. Right
Yeah,
now.
his battery's dying.
Yeah.
All right. Okay. So where was I? So the mold is one of those things. So you don't want to leave things around. You kind of want to prevent dust from being around.
Mhm.
Things like that really help. It's more about when you get mold, you want to prevent it from ever getting to that point. There are fungicides you can try and use. I would do some Google searches on fungicide and algae to make sure that the fungicide does not interrupt with the algae.
Okay.
So, but again, those are good things to learn, best practices in microbiology, how to keep things sterile and stuff like that.
Okay, yeah, so as far as if we were trying to breed the different strands, is there a way to get them, like is there like a certain like chemical or like substance that we could put in that would maybe encourage them to breed more or faster?
I'm not super familiar, but I don't think there's just a chemical, but I think there's a a time in their growth cycle in which they do. So what happens is they form what's called gametes. And those gametes can either propagate into the unicell that they normally were, or they can basically mate with another gamete. You're gonna have to read up some literature on that and how to do it. I'm not sure. I don't think there's a chemical you can just drop in, but I think there's certain periods in the light cycle that they do it. So if you do it at night or something like that, it might help. Things like that. Yeah, but I would think about your experimental design, because it's a very good idea, but it's not the easiest thing to do. But I mean, there's cool things you can do with mutants, right? You can do well. If one mutant makes a lot more lipid, it's usually not universal, meaning that certain points where it can be growing off nitrogen or nitrate or something like that, but they might not do better than a wild type strain, right? If you're at one temperature, it might be less effective in another temperature. The wild type might be more, right? So one of the big problems in the industry is we find all these genes that make more lipid, but when we actually grow it out in the environment, no longer able to compete, and it's very, it becomes detrimental to it. So I would, if you guys do choose to go a different route, is to try and characterize these mutants to see what is it about this gene that makes it make more lipid, but is it consequential in another area, meaning there a temperature that's not good for it, or is there a temperature range or a salinity range, stuff like that.
Yeah.
What we do here at Systems Biology is we want to look at everything. If you look at one gene, you're not going to understand it. So if you look at all the genes, we can kind of get an idea of how this one gene is responding throughout the system, right? It doesn't always work that it's just this is your magic silver bullet.
I have another question. So we did some research and we got to read articles on different types of algae, and one thing we looked at was the flagella and the length of it. I read something about having a longer flagella somehow makes the algae slower. Could you possibly explain that a bit more? It kind of confused me when I read that.
Well, usually, these organisms aren't actually bred or grown for biofuel, right? They're actually living organisms in the ecosystem, right? So the flagella help them be motile in a pond or surface water, wherever they're at. And what happens is if there's too much light, they can start to sink. If there's not enough light, they can start to go up, stuff like that. So having the flagella gives them a set of mobility, right?
Right.
When you remove those or they shorten them, what happens is you're basically conserving energy. Well, that energy can now either go into growth or lipids, whatever it is, right? So just think of like if you prune an apple tree, right? A lot of times they prune apple trees,
Right.
apples, right? And they get bigger apples. So it's the same kind of idea.
So would you say having a longer flagella isn't very helpful when it comes to the lipid content then?
I hesitate to make a general statement like that, but I think that is usually the feeling that if you're growing it in like a reactor or something like that, where it doesn't need to be very mobile, right? You're providing the light for it, you're providing all the nutrients for it. A flagella might be more of a disadvantage.
Okay, thank you.
Okay. So yes.
There are also techniques out there to actually make the algae drop their flagella. I think you can do a pH shock or something like that, and what happens is the flagella will break off. That'd be really cool to see what would happen if lipid accumulation pre and post some kind of flagella shock to see if one makes.
Yeah, I read something about that. I think they also do that with they called it, I think, stressing with like the light levels as well, and it helps them. If you don't give them as much light, it helps them kind of I don't know, be more active is what I read. I believe it was kind of.
That'd be a cool thing to look at. It'd be a cool experiment to do.
Yeah, so just like one follow-up, this is more kind of general, but like as far as like actually analyzing algae under the microscope, how are you able to like see like the flagella and the organelles like easier? Because we can see the cells obviously, but it's like kind of hard to see their characteristics. So is there anything we should look for?
I mean, well, the easiest one for the flagella is you'll see it move around. Right? You'll see it move around. And then you'll like it'll just start swimming, right? It'll just be going crazy. If you want to fix the cells, so what we do is we fix cells. We use formaldehyde. I'm not sure formaldehyde in classrooms.
Yeah, you got something else?
Nah, I don't really. I would probably try high salt. Usually that can shock them.
Put it down, okay.
What was that?
High-
But other than that, you just really need, a pretty good microscope to see organelles and stuff like that. Not much more you can do.
Any more questions?
Questions? No, I think we're good.
Okay, who's
Thank you
next?
so much.
One more group.
Okay.
Last
Good luck,
group.
guys.
Algae fit. Algae fit.
Algae fit.
Yeah. Okay, who are you guys?
I'm Izzy.
I'm Heidi.
I'm Natalie.
I'm Cade.
Hello.
Okay. So for our experiment, we are trying to figure out the best fertilizer using algae to grow butter, lettuce, and spinach. And so our two tests are we were going to add, well, we have like a control, and we were going to add potassium because we read that was like a really good fertilizer that's in a lot of fertilizers to the algae. And then we were going to add CO2 to the algae as well. And then we're going to use it as a fertilizer and then see like which one had the best outcome for the the growth of the plants and the health of the plants. And so
Hold on. So let me see if I can understand your experiment.
yes,
So you can grow the algae basically 3 different ways. One with potassium, extra potassium, correct? What was the second one?
CO2.
So increased CO2.
Yes.
Yeah.
And then the third one was what was the third one?
It just control. Just grow algae and see
So
if
the
it works.
idea, if you grow these algae under different conditions and then use the algae, the biomass as a fertilizer, does that increase productivity of your vegetables?
Yes, that is what we're doing. So one of our probably our main question is that we're having a hard time kind of just like figuring out like the potassium side of it, like how to add it to our algae. And so we talked to one of the chemistry teachers about this, and she said the best way would be potassium chloride. But we're wondering if that is gonna like harm our algae.
Because she's saying that it's like a salt, and so we don't want to like add too much. But we
Correct.
don't know how much
Is
too much.
Yeah.
Okay, so one thing to think about is what you guys are basically trying to say is, can we store this molecule in the algae and then use that as a fertilizer, right?
Yeah.
Usually algae don't store potassium.
Okay.
Your problem is going to be, how do I get a potassium enriched algae that I can use as fertilizer? It's not necessarily about adding more chloride or potassium chloride. It's more about increasing the amount of potassium influx into the algae. Now they have, and I would look up for you guys, is they have potassium ion channels. There's potassium proton channels. Look at those and see what they're doing. Usually those are used to balance a proton motive force across the membranes. Thanks, Josh. Those are really important to maintain osmolarity within the algae, okay? So you're gonna have to go across a barrier to get more potassium in. And I think probably your best bet would be grow that algae at different pHs. The problem with that is you're not going to really be able to assess how much potassium went into the algae.
Okay.
Right? That's not something that you can just do readily. The CO2 one's obvious, right? That if you have better growth, more CO2 in the algae, you can assess that by how fast it grows, how much it grows, if there's any lipid, that kind of stuff. So that one's a good way to go. The potassium, I'm not sure. Maybe you should try something like nitrogen. Right? Nitrogen's always good for plants. Phosphate's always good for plants. Right? So what you can do is you can do an experiment where you try, look at, if I give it ammonia versus nitrate, what's the difference? If I give it more phosphate versus what? So the algae will store nitrate and phosphates often. They store nitrates usually as putrescine or polyamines. Also, they store it as a lot of their amino acids are all filled with amine groups, and so they're more readily available to store nitrogen or phosphates. So I would look into that.
Okay. That's good. Thank you. So then you already talked about the CO2 with the other groups. We got that down. Should I put Bold's medium?
Yeah.
Okay, so would Bold's medium affect the soil and the growth of the lettuce?
Yeah, I think it would, right? There's nutrients in there. Were you gonna like filter out the algae before you added it to the soil? Or
We weren't thinking we would, but we could. I don't know. That was something.
we were kind of debating over if we should dry it or we should just like.
It'd be easier just to pour it in, but
okay, so if you were gonna pour it in, what would be one control you would have to do?
what? Sorry,
Control.
say that again.
Right,
I didn't
control.
hear that.
Yeah, if you were gonna pour in your algae that has the media that's in the media, what would be one control you would have to do for your plant?
You'd have to pour in like without algae, like the bulbs,
Right.
In a different plant.
Exactly, right? Because there's going to be nitrogen, phosphate, there's going to be all kinds of stuff in that media that's actually good for the plants. I mean, algae is pretty much a plant itself, right? So if it grows well on bulbs, what do you think the plant's going to do really well? The idea with using algae as a fertilizer is that it comes with all the carbon, and the carbon can be consumed by microbes, and the microbes can feed that to the plants, right? It's not that the plants are actually growing on the algae.
Yeah.
Right? You understand that, right? So
Yeah.
what you're trying to do is grow, and that's the same reason people use manure, right? Manure isn't for plants to uptake; it's for the microbes. The microbes break that down into ingredients that the plant can take up: the nitrates, the phosphates, that kind of stuff.
Mhm.
So what you're trying to do is create a fertilizer that can be broken down by bacteria that can then be used by the plants more readily. So think about that. So I would focus more on. I would filter out the media, so you're
Okay.
just doing biomass and see how that works.
Okay. And then should I ask this?
Will the added phosphate and what is the one nitrate? Will that
Yeah.
harm the plants or the people who eat the plants if we add it to them?
Definitely won't hurt the people because it'll stay in the soil if it's too much. You can get nitrate burn, so if you have too much nitrate in your soil, it can burn the roots and the plant doesn't do very well. No, when plants uptake nitrate, what they do is they take that nitrate, they put it on their amino acids, and they make protein out of it. So it's basically just protein, or it's just a nutrient for the plant, and that's something that we need, right? So we can't build proteins without amino acids, and amino acids are nothing without nitrogen. So it's a little bit different.
Okay, and then since we shouldn't do potassium, so what? Sorry. Since we're not doing potassium, how much phosphate or nitrogen should we put in?
I would see what the Bold's recipe is, right?
Yeah.
Because you got to start there because if you're using Bold's already pre-made, it already has a certain amount of phosphate in it. If you make your own Bold's, you would want to test different amounts of phosphate and base that off of what the Bold says. Whenever you get into like the millimolar of phosphate or nitrate, that's starting.
I think he froze.
Did he go away?
He's like,
He's right there.
but he looks frozen.
We think you froze.
Sorry, you guys still there?
Yep,
Yeah.
we're there.
I don't know what happened. Where was I? I was talking about nitrate or something, right?
Yeah.
Or phosphate. Just whenever like 1 or 2 millimolar is plenty phosphate for algae.
1 or 2 millimolar right there.
Okay. Got it. Okay, are we good?
The other thing, I know you guys are thinking about CO2. One thing you can do, remember we talked about what does CO2 do when it dissolves in water? Try making bowls without adding a balloon of CO2 or anything, and just say with bicarbonate, right? Because now instead of giving it CO2 as an air form, a gas form, you're actually giving it the chemical bicarbonate.
Okay.
And see if there's a difference between your growth. And what you should do is you should do some calculations on the amount of CO2 within that volume of balloon, and add the same amount of CO or bicarbonate to a flask that's covered. Does that make sense?
Yeah.
Now you're assessing the difference between CO2, atmospheric CO2 on growth versus the chemical structure of bicarbonate and by adding it into the water, into the media. That'd be a really cool experiment also.
Yeah, that'd be a cool experiment too. Nice to find out. gas versus solid.
Yeah.
Okay.
Okay, well, I think we're
Thank
good as a
you.
group. Yes.
Yeah.
Thank you.
Thank you.
Good luck, you guys.
Bell's gonna ring, so thanks, Jake, and we'll probably need more help next week.
Yeah, let me know. I'd love to do it, and I'll definitely come by for the night.
Yeah, that's May 22nd. I'll send you an email with all the dates on that. That's gonna be in the morning, like a Monday morning.
Yep.
All right. Hey, thanks. Talk to you soon.